In the first installment of this series on the Twilight of the American Empire, we saw how several different scholars, using different methodologies, operating from within different disciplines, and writing at different times, all point to a coming crisis for the American Empire in the late 2020s or early 2030s. And, taking their analyses in total, it appears reasonable to predict this crisis will occasion the collapse of the American Empire. I want to follow up this analysis with a deep dive into how these developments, and the empire rise and fall cycle generally, is explained by a deep understanding of evolutionary biology. However, I appreciate that not everyone will have the depth of knowledge of evolutionary theory necessary to follow that discussion, so this second installment will serve as a background primer for those who might benefit from it. Though there will be several things I'll explain on the way there, the primary destination for the exposition in this second installment will be fleshing out the difference between the fitness-enhancing selection processes, which I'll call here genetic natural selection and phenotypic natural selection. If you're confident you know the difference between these two references, you should be fine for skipping over this video and going straight to the third installment of the series. I'm going to argue that it is evolutionary biology that provides the ultimate explanation for the empire cycle. However, since both Glob and Turchin identify this cycle as being about 250 years, this would seem to fly in the face of conventional Darwinian thinking. We know that the standard, modern evolutionary model operates at far slower rates than that. While evidence has emerged that even this model of evolution, based upon fitness-enhancing traits emerging from genetic mutation, can act far more rapidly than once thought, having important impacts over mere millennia. Despite this, though, no one suggests that such change can happen over mere centuries and presumably, to explain the cycle, somehow cycle back. New mutations taking the organism back to what it was before. But the process I'll be describing is indeed a form of natural selection, selecting for fitness enhancement, just as evolutionary theory would predict. The confusion here arises from using the phrases natural selection and fitness enhancement without recognizing that they both apply to different selective processes. As I've already hinted, the two emphasized here are the natural selection of genes and phenotypes. In fact, though, there are three kinds of selection processes that need to be acknowledged to be clear about the argument to be made here. These three are genetic, epigenetic, and phenotypic selection. The situation is even further confused by the fact that all these processes are described as taking place in the context of something called the environment. However, this word environment means quite different things in these different contexts. So to understand what's going on with sufficient clarity to appreciate the argument to be made about the role of natural selection within the empire cycle, we'll have to unpack all these ideas and assure that we're on the same page. Let's start then by clarifying some concepts. The first of these, which sometimes confuses people, is the concept of fitness. Some people, even scholars, think that fitness refers to survival, as in the phrase survival of the fittest. Along similar lines, some think it assumes the organism with the greatest fitness will be the strongest, or the largest, fastest, or whatever. None of this is true. Fitness refers exclusively to an organism's success in transferring its genes into future generations. In fact, to be precise, it is more the genes or the genome than the organism which is fit. That is, able to produce an appropriately sculpted phenotype for sustaining its own immortality. A long lifespan or great physical strength provide no fitness advantage if they do not cash out in effective reproductive success. Fitness is only a measure of such reproductive success. A second core, but frequently misunderstood, concept is the phenotype. I'll assume that anyone who's gotten this far is already familiar with the concept of the gene, or the genome. The genes of a genome create a phenotype. The process of this creation is called ontogeny. A genome has the potential to create different kinds of phenotypes, and an obvious example is the ability to create a male or female phenotype. But depending on the species, there's a range of other possibilities too. For example, the genome of the speckled wood butterfly can create two kinds of males. These two are distinguished by both morphological features as well as different mating strategies. These are defenders or patrollers. Defenders find sunny areas that attract females and defend them against other male defenders. Patrollers, though, make no effort to take over such territory and travel about looking for available females who have at the time in their short mating life defined a defender's territory. The two phenotypes are not only distinguished behaviorally but also morphologically. The defenders have 4 spots on their wings while patrollers only have 3. However, the proportion of each phenotype is influenced by the environmental conditions. At times and in places where there are more sunny spots in the forest, there are more defenders with their 4-spotted wings. When there are fewer sunny spots, there are more patrollers with their 3-spotted wings. Somehow, the speckled wood butterfly genomes can gather and integrate information that mediates the frequency of each male genome appropriate to fitness-relevant circumstances in the world. This is an example of epigenetic selection. It is part of the autogenetic process, the development of the phenotype. A useful way to understand what is happening here is to clear up the confusion about the use of the word environment in both the evolutionary and autogenetic context. In the evolutionary context, environment refers to the objective material world with all its forces, including the existence of other forms of agency which may interact with an organism in any of a variety of ways. For example, mating, cooperation, competition, predation. Any genetic mutation that provides a trait which allows the organism to better leverage any of these interactions so as to improve its fitness is effectively selected for by evolution to be perpetuated into the future lineage of the organism and the species gene pool. This is natural selection of the gene. In ontogeny, though, while the same word environment is used, this refers to a very different process. In ontogeny, the environment is better thought of as a field of information, and, indeed, rather than thinking of it as a material world of forces, including forms of agency, this field of information, in a certain sense, is created by the genome, or at least the genes, in the autogenetic process. This is where so many people get confused. I discuss all this at great length in my book, Biological Realism, which I'll link in the show notes, and strongly recommend if you want to understand all this stuff more thoroughly than I can explain it here. Basically, the genes of the genome have been molded by evolutionary selection to build mechanisms, perceptual, cognitive, and behavioral mechanisms, which scan and probe that field of information called the environment for input that will influence the specific form of the phenotype being created. While, as far as I know, we can't identify the exact mechanism, this explanation explains how it is that the proportion of male speckled wood butterflies, which are either defenders or patrollers, seems to be determined by the prevalence of defendable territory that's available. Somehow, in the autogenetic process, these perceptive and cognitive mechanisms created by the butterfly's genome extracts the information to guide which strategic phenotype to produce. The same processes are observable in humans, remembering that autogenetic refers to the entire developmental process all the way to becoming an adult. An obvious example here is the impact upon offspring's sexual strategies of those aspects of the informational environment constituted out of the early childhood home experience. Children's genomes that experience high marital discord, high stress, and inadequate resources are more likely to generate faster sexual strategies, including early onset of puberty, early and precocious sexual behavior, emphasizing short-term relationships and reduced investment in offspring. In contrast, marital harmony and adequate resources tend to produce slower sexual strategies, with later onset of puberty and sexual experience and an emphasis on long-term relationships and higher investment in offspring. Of course, it is important not to overstate the autogenetic process here. Behavioral genetics has shown that these strategies have a significant degree of heritability, in which such dispositions come directly from the parent's genes. However, even when such heritability is in the 0.45 range, that still leaves much of the variance to be explained by these autogenetic processes of reading the environment. And of course, on average, these outcomes are fitness enhancing. In a social context where resources are scarce and parental bonds are vicarious, a faster sexual strategy would be more fit. And likewise, the opposite, when resources are not scarce and parental bonds are durable. So ontogeny, just like evolution, gives rise to the kinds of phenotypes that are available to specific genomes. Evolution must mold these autogenetic options, but once they exist, the range of potential phenotypes is expanded. The final preliminary term to be introduced here, before the distinction between genetic and epigenetic and phenotypic selection can be parsed, is the concept of the reaction norm. A reaction norm is a conceptual space. When applied to an individual genome, it is the space that captures every possible phenotypic variation of which a genome is ontogenetically capable of producing. The term reaction norm, though, also can be applied to a species gene pool. Within the same species, different genomes have different reaction norms compared to each other. For instance, in most sexually reproducing species, and there may be some weird outliers like among fungus, but usually, a specific genome will only produce either a male or a female phenotype. And there could well be other nuances. Between different genomes, there may be variation in the existence or sensitivity of the perceptual or cognitive mechanisms that regulate ontogeny in relation to the informational environment. So a species reaction norm captures all the phenotypic possibilities possessed by all the genomes of that species. Any imaginable phenotype, which no genome in the species can produce, is outside of the reaction norm of the species gene pool. This is how I'll use the term reaction norm going forward, referring to the set of possible phenotypes for a species, specifically, of course, the human species. Now, with all these terms clear, it's easy to explain the distinction I set out at the start between the natural selection processes of genes and phenotypes. Genetic natural selection, through the introduction of new phenotypic possibilities created by genetic mutation, lands outside the species' pre-existing reaction norm. Effectively, this mutation, through genetic natural selection, shifts the boundary of the species' reaction norm. And of course, ever so slightly shifts the very nature, therefore, of the species. Given enough time, with enough such subtle shifting, a gene pool can see its reaction norm shift so far away from a designated starting point that the phenotypes in the reaction norm space are so different that that gene pool can no longer be said to belong to the same species. This is what we mean by Darwinian evolution in the standard sense of that word. Epigenetic selection, then, using the opportunities made available through genetic natural selection and the mechanisms for monitoring the informational environment of ontogeny, gives rise to a variety of phenotypes. If genetic selection has provided the requisite phenotypic flexibility, it can be expected that the specific phenotypes generated through epigenetic selection will be on average the best selected phenotypes for the organism's environment. However, just as epigenetic selection evolved to deal with environmental variability that was too changeable for genetic evolution to keep pace, so it can be that environmental changes can outpace even epigenetic selection. In that situation, phenotypic selection becomes the best available option for the gene pool. Unlike genetic selection, which defines the existing boundaries of the reaction norm, or epigenetic selection that purposefully redefines the phenotypes available within the reaction norm boundaries, Phenotypic evolution makes the best of a sudden new environmental challenge by selecting among the available options within the existing set of phenotypic resources. This can be visualized as shifting fitness focus toward other regions of the reaction norm where more optimally fit phenotypes are located. While the process of fitness-enhancing natural selection is involved, unlike in genetic selection, phenotypic selection provides fitness enhancement without redefining the boundaries of the species reaction norm. Unlike epigenetic selection, phenotypic selection provides fitness enhancement without redefining the nature of the phenotypes available. Perhaps the most famous example of this is the pepper moths in England during the Industrial Revolution. Prior to the Industrial Revolution, this moth was known for its mottled white appearance. As a nocturnal hunter, it spent its days sleeping on tree trunks where it could effectively blend into its background. During the Industrial Revolution, though, great amounts of soot was dispersed across the region, covering the trees upon which the pepper moths slept out their days. Now, though, against the blackened background, their mottled white wasn't nearly so effective a camouflage, and they were much more vulnerable to predation. Genetic researchers now believe that around 1819, a mutation in the peppered moths started producing black phenotypes of the species, which could outcompete the white version through better daytime camouflage. This was a case of genetic natural selection, which moved the boundaries of the species reaction norm to include a previously non-existing phenotype. However, the original mottled white phenotypes did not go extinct. And in fact, once the industrial pollution was dramatically reduced around urban England in the 20th century, the camouflage advantages of the original mottled white phenotype returned, and it came back with a vengeance. This was phenotypic natural selection, in which the boundaries of the reaction norm did not change, but the fitness-enhancing location within the reaction norm shifted back to the white phenotype of the peppered moth. Changing conditions can change the boundaries of the reaction norm, but it need not do so to generate a form of natural selection that promotes specific phenotypes depending upon the shifting conditions of the world that the species and its gene pool confronts. What I'm going to argue in the next installment of this series is that it is just this kind of phenotypic natural selection which explains both the cycle of empires and why there's likely little to be done to avert the probable arrival of the American empire's decline. Which is not to say, of course, that you can't take action to prepare yourself for that historical event. But all that we'll understand better as we start our next discussion in the following video, and I'll see you over there.